Okay, thank you, JJ. So I'm here to talk about rapid analytical methods that we've developed as part of the Dibanet project. This is part of the second work package of Dibanet. We have several tasks involved in that. And the first one involves finding the best feedstocks for producing levulinic acid via acid hydrolysis, both in Europe and also in Latin America. So we would identify which feedstocks have potential, particularly for acid hydrolysis purposes, and we'll analyze these via wet chemical methods and determine the most optimal feedstock for the process. There's three main partners in this work package. There's the University of Limerick, which is responsible for looking at European feedstocks. There's CTC in Brazil, which is responsible for sugarcane bagasse and sugarcane trash, the field residues. And there's Unicamp, also in Brazil, and they're looking at other biomass in Latin America. Here we have some of the different feedstocks, both in Ireland and in Brazil, that we looked at. The ones that I've underlined here, that we developed near-infrared spectroscopy models for their quantification. You can see there's a wide variety of different feedstocks, ranging from energy crop, like miscanthus, to municipal wastes and agricultural residues. What's important? Well, if we're doing a chemical hydrolysis, like in the Dibanet process, the most important properties are the polysaccharide sugars. In particular, glucose, which is mainly in cellulose, and the pentose xylose, which is in hemicellulose. And from that, we will get furfural. Also important is the lignin content, because this affects your acid hydrolysis residues, and other properties such as the ash content. Here's a summary of the wet chemical analysis that was conducted at UL, CTC, and Unicamp for the main feedstocks as well as other feedstocks. You can see we've done a huge amount of analysis over the course of the project, from which we have got very significant results. The early evaluation at the University of Limerick involved looking at the most suitable feedstocks. We identified miscanthus at the very early phase as being a very good feedstock based on its productivity and low production costs and good environmental profile. We also looked at various waste materials. Cardboard and paper are particularly useful due to their very high cellulose contents. You have waste materials such as animal slurries, but these, although they can have a decent sugar composition, they are very wet and their transport costs will be significant. What was quite interesting is that if you look at the literature that's out there, you see a lot of data, but a lot of it is based on very poor, very outdated analytical techniques. So spent mushroom compost has been identified in the past as a suitable substrate for biorefining. But this is based on acid detergent fiber methods that indirectly detect these polysaccharides. What we did is we directly detected the sugars that are present within them, and we found that they're not there. It's a misaccurate prediction using these detergent fiber methods. We only found a maximum of 20% carbohydrate, which is not enough to warrant processing in Dibanet. We also looked at municipal wastes. Waste food is not suitable due to its low sugar content and low cellulose content. Green waste can have significant cellulose contents, but it depends what type of green waste you're talking about. If you're using woody material, it's of value. If you're using grass and foliage, generally there's not enough carbohydrate in that material. So from the University of Limerick, we concluded miscanthus is the best feedstock to focus on. We also investigated cereal straws and waste paper materials. Unicamp in Brazil were given a total of 10 different feedstocks to investigate via wet chemical methods. These are all generally waste materials from the agricultural industries. Here are some compositional data obtained for these samples. Of most interest would be the total sugar content, and we're looking for a high value here, providing that the feedstock can be still obtained relatively cheaply. On the basis of the result, and also on the basis of a very thorough examination of environmental costs, supply cycle levels, and transport costs, they decided to investigate these three residues: banana, coffee, and coconut. And so what we found in our early phase is that there's a number of viable feedstocks to utilize in the Dibanet process. A lot of the secondary data, analytical data, was very poor. So it's very important for us to do our own wet chemical analysis and to release this data to the general public so that they can also use it in their own schemes. So what we did is we detailed evaluation of a number of feedstocks by wet chemical techniques. We developed in Limerick analytical methodologies which you can download from the Dibanet website, and these were used by each partner, and we had very strict quality controls on our work. So here's one of the procedures you can download and the data recording sheets. The result of this analysis is that we have a database that you can download from the Dibanet website containing compositional data of 1,281 samples from Europe and Latin America. There should be a video. So if you go to the website, you can download this program. It gives you a list. Here you have the 1,200 samples. You have summary statistics of their compositional range, max, min, standard deviation. These are the different types of materials. If you double click on the sample, you can see more detail about its compositional characteristics. You can see the predicted yields per ton from different biorefining processes, this one being Dibanet. And you can also filter according to your desired feedstock. So here we're looking at waste materials that you can find in Europe. And once again, I'm double clicking to get the analytical data for that. Another output of the project is we developed a report summarizing the findings of our research. We have a 150-page document that you can download from Dibanet. If you don't like the sound of that, there's a 16-page version that you can also download. So in University of Limerick, we looked at miscanthus. It's a very productive energy crop, and it has a very attractive composition for biorefining purposes. We didn't just take a whole crop, we took different parts of the plant and analysed these to see their relative value. So we took the stem, different sections of the stem, leaves, the leaf blades, the leaf sheaths, and we found that the composition varies significantly between these different fractions. For example, the stem has a higher total sugar content than the leaves, whereas other things in the leaves are greater, such as your acid-soluble lignin and your extractives and your ash. These are generally not good properties for hydrolysis, particularly chemical hydrolysis. We sampled miscanthus plants from different plantations that had different years of productivity. For example, in the first year you get very poor growth, and it's not recommended to utilize that material in a biorefinery, both based on its total sugars content and also its productivity. Instead, you should wait to subsequent years to collect the biomass. Now, miscanthus can be harvested between October and April, so you have approximately a 6-month period for collecting the crop. And over that 6-month period, we sampled from 7 different sites each month a number of miscanthus plants, and we looked to see what is happening both in terms of the relative proportions of the different fractions of the plant, leaves versus stems, and also in terms of the composition of that plant. And what did we find? Well, we found between October and early December, not much of the biomass is lost. It's relatively green, and you still have the leaves on the plants. We term this the early harvest period. Between then, December and the end of February, you have a rapid loss of leaves from the plants. The harvestable biomass decreases significantly. Once you get to March, most of the leaves have dropped. You only have some that are left, which are the sheaths attached to the stem. And these are lost much more slowly. So we call this the late harvest period. And we found that the best time for harvesting miscanthus is dependent on what you're going to do with the crop. In the early harvest, you have approximately 30% more standing biomass per hectare than you have in the late harvest. So if your only focus is biomass yield, harvest it early. However, you have a 50% moisture on a wet basis from this early harvested crop. And that is not going to be suitable for most thermochemical processes, such as pyrolysis and gasification. But an early harvest is feasible for the Dibanet process and for a lot of other hydrolysis processes. Now, this early harvest will remove leaves that would otherwise be left to the soil, so you need to consider addressing the loss of fertilizing value by adding fertilizer to the soil to cover that. There are also significant changes in the composition of the biomass over the harvest window. On a dry mass per ton basis, the biomass collected at the end of the period has a much higher total sugars content than the material that's collected early. Here we see a plot showing the dry leaves content as a percentage of the stem weight. And you can see that at the very early period, this is the early window here, your leaves weigh approximately half as much as your stem mass does. At the late harvest period, you only have the sheaths that are left attached to the stem, and you have much less of these. So you see a significant loss of biomass over that period. What we've done here, on the right axis, you see the biomass yield in terms of dry tons per hectare. We've assumed that stems remain as harvestable biomass throughout the course of the harvest window, this 6-month period, but the leaves are lost. So what you see is that you have 12.5 tons per hectare at the late harvest. This rises to 16.5 tons per hectare in an early harvest, so significantly more biomass. What happens to the composition? This is your pentose content, there's no change. This is your hexose content, it goes up, and that is because you have more cellulose in the stems than the leaves, and over time you have more stems than leaves. So then we use that to plot the linoleic acid yield through the Dibanet process from 1 hectare of biomass. So you're linking the change in standing biomass with the change in composition. And you see the relative advantage of harvesting earlier compared to harvesting this last period. And you can see you can get up to 20% more biofuel or more platform chemicals if you harvest early compared to when you harvest in the latest period. Here's just some summary of the composition of 1 ton of biomass harvested. The late is the red, and the early period is the blue. You can see that in the later period, place on lignin is higher, glucan is higher, xylose does not significantly change. For the minor components such as ash, extractives, acid-soluble lignin, nitrogen, galactose, and everything here, you have higher composition of values for the early period compared with the late period. So there's another video here. This is another database that you can download from the Carbolia website. It gives you a list of the miscanthus plants that we collected over 1 season in 7 different locations. These are the locations. And you can filter this according to location. So in this case, I've selected 1 particular stand and 1 particular modal height class of plant. I press the filter button and we get 11 plants. And we can see the trends that occur over that period. So here we see the loss of the leaf mass from the start to the end of the harvest window. Here we see the effect on the glucan content from the start to the end. It's going up. Galactose is going down. Place on lignin is going up. Acid-soluble lignin is going down. Here you see the yields per ton from biorefining processes, and here are the yields per hectare from the Dibanet process, for example. And using that list, if you double-click on a sample, or plant, you see more detail about the composition on a whole plant basis, on a stem basis, and on a leaf basis. You can see the individual plant fractions, the mass proportions that they constitute to the plant. And if you click on one of those fractions, you can see the individual compositional data for that. So here we have the leaves of the plant, moisture data, elemental data, and predicted yields from processing it in a range of processes such as the Dibanet technology. Okay, we move on to sugarcane residues, which is the work of CTC. Here we have sugarcane being harvested. What that does is the stems are chopped and put into this trailer. The leaves and the tops of the plant are left on the floor. This is called sugarcane trash. The stems then go to a sugar mill. They're processed. You get the sucrose from the sugarcane, and you're left with bagasse, which is the fibrous residue. And that is much in excess of what is needed to process the millet. And here you see huge mounds of bagasse, and there's hundreds of millions of tons produced in Brazil that can be used for biorefining purposes. Here's just a slide showing how the production of sugar, alcohol, and sugar cane has increased significantly in Brazil since the start of the pro-alcohol program in 1970. What we did at CTC is we focused on the Sao Paulo region, because this is where most of your sugarcane plantations are. We sampled the gas from a variety of different sugarcane mills in that state. And then we did analysis of these samples. And here's just some summary data of the compositions that we found. In particular, what's important is when you're processing it is the glucan and the xylan content. And here we see there's some variation. However, a lot of the variation is on a ash-free. On a whole mass basis is 10%, and on an ash-free basis is 5%. So a lot of variation is due to the ash content of your bagasse, which can vary significantly between days and also between different mills. We found that both samples, bagasse and trash, have enough sugars to warrant processing in biorefining processes such as Dibanet. Bagasse varies, and particularly the ash content varies. And it's very important that you analyse this over a period of time, because ash can be a significant problem in some processes, particularly thermochemical processes. And you may need to discriminate between high and low ash contents for suitability for processing in technologies such as Dibanet or pyrolysis. We did crude estimates based on the arisings, the productions of the gas and straw in Brazil, and we found you can have massive yields potentially from the Dibanet and other biorefining processes. You can significantly increase the production of diesel or gasoline miscible biofuels by utilising these more efficiently. Now, at UniCamp, they analysed coffee. They sampled several parts of the plant, the leaves, the husks, the grain, and they also did some ground coffee analysis for comparison. Various kinds of varieties of coffee plant were also sampled. Here are some of the histograms for the composition. This is clay-on lignin. This is ash. You can see there's quite a significant variation in the composition between all the samples that were analysed. Banana, then, we sampled a whole variety of different parts of the plant, the leaves, the stem, the rhizome and the stalk, as well as the peel itself. And we have some more histograms. I'll move to the next slide because, okay. So you see once again the clay-on lignin content and the sugars content as well. They all vary quite significantly. And we determined that different parts of the plant, some have value and some do not. So not all of them could be economically processed in a biorefinery. Here we have coconut samples. Two parts of the plant were analysed, the husks and the fibres. Once again, we have some histograms. Here's an important one. It's the glucose content. It's ranging by approximately 20%. And that's a huge difference in terms of your potential yields. 15% is not enough to warrant it in the Dibanet process. 30% when you're considering xylan and other sugars is enough. So it's very important to look at the specifics of your feedstock. For example, once again, total sugars content, that varies 20%. Okay, the next task we involved with was developing lab-based near-infrared spectroscopy calibration equations. Now, near-infrared spectroscopy, in case you don't know, it involves the absorbance of near-infrared radiation by biomass samples. And from these absorbance spectra, we can determine via chemometric techniques the compositional data for those samples. For example, the glucose content, the xylan content, and so on. So in Europe and Latin America, we developed models for 5 different feedstocks, and here are the responsibilities between the partners for these. Why do we need to do this? Well, if you analyse via conventional wet chemical methods, it's a lot of work. It's a lot of brain-numbing monkey work. You chop the sample, you have to mill it, you have to air dry it. It's very, very boring and laborious. What we want to do is have no work involved whatsoever except scanning the material. The minimal, if we have to do something, all we want to do is really to chop it up and then present it to the analytical cell of the NIR device. But we want to do this with a wet, chopped sample. And all of the literature that's out there is based on something such as this. A dry material that has been air dried over several days, ground and sieved to be it's very homogeneous. And this is taking away the whole concept of rapid analysis, because this is several days of work, or at least several days of waiting until it's ready. So what did we do? We did a scan at several stages along the sample preparation methodology. You have a wet on ground material, so that the material in the red box in the previous slide. We call this a WU scan. We air dried it, so it was dry, but it was still about heterogeneous particle size. This was our dry on ground scan. We ground it down for a dry ground scan, and then we sieved it to a very homogeneous particle size distribution, which we called our DS, or dry sieve scan. And logically, you would have the best models for predicting glucan, cellulose, clay-on lignin by using this scan, and the worst by using this scan. But we wanted to prove that these could still be very good for quantification. And here you see comparisons. This is one sample and several different scans. And the one that stands out is this blue one. and that's based on the wet on-ground material. You have all these little undulations here for the dry samples. Some of these are not present with the wet material, and that's because moisture is very strongly absorbing of NIR radiation. And people don't like to use this kind of material because they think there's not enough detail to make a model and to make a prediction. But we showed that you can do that. Here you just see a number of samples that are from the wet on ground, the WU spectra for miscanthus. You can see there's a huge variation according to the different sample types. How do we test the accuracy, the quality of these models? Is we build a model based on a calibration set of samples. And then we test them on an independent validation set of samples that are not used to develop the model. And we give standard statistics such as R-squared and standard error prediction, so it's your standard error in what you estimate the compositional values to be. And a nice technique for comparing the quality of models is to take your ranging composition and divide it by this standard error of prediction. And there are guidelines out there. If you have over 15 for this RER ratio, you have a model that's suitable, that's very good for quantification purposes. Payment schemes, for example. If between 10 and 15, you can use it as screening control, and between 5 and 10, you can use it in rough sample screening. So here we have some statistics for the work at University of Limerick on miscanthus, and this is for glucan, so predominantly the cellulose content. Here's your R-squared for the prediction, for the validation set, for the dry sieve material. Very high R-squared. Your standard error prediction is 0.8, 0.9%. And you have very high RER ratio. However, this is what we're really interested in, is the wet on-ground sample. And you still have a good R-squared, a very good R-squared, and an accurate standard error prediction. And your RER ratio is over that 15 threshold. And here you see a regression plot. On the left-hand side is your DS model based on the dry homogeneous samples, and here is your wet on-ground model. So while this one is closer to the line, this one still gives you a very nice fit. I'm not going to go through all the models, but what's the important ones? Glucan, xylose, because xylose is about 25% in miscanthus. Once again, you have good R-squared prediction, and a low standard error prediction, and RER ratios that are over 15. The same is true of place-on lignin, which is another 20 to 25% of your sample. Look at this with the WU. We have an R-squared of 0.96 and a standard error of about 0.6, so we have very good predictions. Here is a summary for miscanthus for all the different constituents that we calibrated for. Even minor components can be predicted, such as arabinose, mannose, and rhamnose, that are very small concentrations in miscanthus. They still have good accuracy for most analytes, for the DS in particular, but also for the wet on-ground spectra. We have a lot of good predictions from that. There's a paper about this that I wrote that you can download from the Carbonia website. We developed many other models in University of Limerick. I'm not going to talk about these. One of them is about pre-treated miscanthus, which I believe Donica talked about, the pre-treatment process that we developed at University of Limerick. What you can see here is that there's a very wide variation in the glucan content of these samples. This is untreated miscanthus, and these are all treated samples. And you have a range of around 40% for the glucan content. The spectral variability is very high, and yet we can still predict very well across this range. Unicamp developed a wide variety of different models for banana, which is the B, coffee, which is the C, and coconut, which is the COC. And you can see that they had good R-squared, both for validation and calibration, and low standard errors in their predictions. Both for the DS, the dry sieve material, for the dry on-ground material, and most importantly, for the wet on-ground material. Once again, you have a high R-squared of validation. And most importantly, you have the sugar models, and once again, you have good levels of prediction. For example, in coconut, you have an R-squared of 0.94. Here we see some of their regression plots. This is for Clayson ligand, once again, a nice fit over a very wide range, which is nice. What's quite interesting, this is for banana. Banana has, on average, a much lower lignin content than a lot of other lignocellulosic feedstocks, which could mean it's very useful for processing in a biorefinery. Coffee residues, once again, this is the lignin content. You have a nice fit and a nice spread of these samples. And coconuts you have here, total sugars ranging from 30 to 52%, and we can predict these quite accurately using the models of Unicam. In CTC, we develop models for sugarcane trash and sugarcane bagasse. Here we see, based on lignin content, we have nice R-squared of prediction. Given that our variation, our range in the composition is quite low, we still have are able to get a good R-squared. Same for extracted content and for ash content. And we've done it also for sugarcane bagasse samples. We found that we need to work very hard in developing models for sugarcane bagasse because the ash varies so much, and the ash is an interference in near-infrared prediction. Yeah, something about the ash content. Each one of these is a mill that we sampled from, and here you see the variability. These are quantiles plots for each mill, and on the y-axis we have the ash content. So if you take this mill, very little variation, very low ash content. If you take this mill, wide variation, up to 15% ash content, which can be very significant. So just to summarize the amount of work that we've done, we've had a huge amount of samples collected and spectra taken for these samples, up to 10,000 spectra over the course of the project. One of the major outputs of this is a company that we have launched at the University of Limerick, the Biomass Analysis Centre of Ireland. This is going to be launched very soon, and we will offer two services: the wet chemical analysis for lignocellulosic constituents and the NIR analysis. And we found that when we compared the quality of our research to other papers that are out there, we have much higher accuracy in our wet chemical methods, and that allows us to offer very good guarantees on the precision of analysis that we can offer to potential customers. We also have near-infrared spectroscopy analysis. We will predict all these different constituents, such as glucan, xylan, Clayson, lignin, ash, for a wide variety of samples, using either specific models like from Miscanthus or global models based on all the samples that we have analysed. Let's skip this. And what's important is that we developed a business plan for the company, and we plan to continue and strengthen the ties that we have between the EU and Latin America by working continually in the future with CTC and Unicam. We have estimates of price. If you're interested, you can talk to me about that later on. Other outputs of the task, we're able, using near-infrared spectroscopy, to discriminate between samples. So we could do qualitative as well as quantitative analysis. For example, with Miscanthus, we can discriminate between a sample that was collected early in the window, harvest window, and late in the window. We can discriminate between varieties of Miscanthus based on Giganteus, which is the major commercial variety being grown in Europe, or other varieties. We can discriminate between plant fraction, stems, internode, leaves, live leaves, dead sheaths, and so on, and the age of the plant according to whether it's in its first year of production or subsequent years. We also have used NIR and other chemometric techniques to analyse in a rapid means products from the process in work package 3s, what Donica was talking about, the acid hydrolysis and the pre-treatment of biomass. So the acid hydrolysis residues, pre-treated Miscanthus, analytical hydrolysates, the liquid hydrolysates from work package 2 and work package 3. And here you see, this is the output from our Dibanet reactor. We have developed a means instantly via transmission ultraviolet spectroscopy to determine the hydroxymethylfurfural content quite accurately. And this helps us when we don't have time to use the chromatography system, or when that's occupied or not working. We can get good estimates based on instant analysis using ultraviolet spectroscopy. This is, again, for the total hexose content. We can predict that also using ultraviolet spectroscopy. The next task, we involved transferring these NIR methods to an online facility at a sugar mill in Brazil. This is a basic representation of a sugar mill. You put your sugar cane in here, it's shredded. You can have an NIR system analysing the shredded cane before it goes into the mill. You then go through a series of mill rollers that extract the sucrose from the cane, and what you're left with is this, which is the sugarcane bagasse. And we have systems for analysing this bagasse in real time using NIR devices. So what we did early on is we installed a system in an older system in a sugar mill in Quatá, Sao Paulo, and we monitored its performance. And there should be a video here. Here you see the conveyor belt of the gas, and you have the NIR device above it. The problem that you have here is the gas is the level of the height is going up and down, and you're not giving a consistent sample presentation method to the system. These are the spectra that we obtained in the laboratory for wet the gas. These are the spectra that we obtained from the online system. There's a lot more noise and a lot more variability. And a lot of that's due to the variation sample presentation. And you had a lot of frequent spectra that weren't the identical fibers anyway. And we found when we tried to do even the basic compositional parameters such as moisture, the R squared for validation was very low. This is not a good system for determining either moisture or the pole content of the gas. So then we tried again. We've got a much improved system, which is called the ProFOS system, and it uses diode array detection. So there's no moving parts. It's much more durable in the environment of a sugar mill. This was installed this year in August. Here you see the photographs of the ProFOS system. This is the bagasse chute, and this is the hole from which you have the lens of the NIR device. Here you see a video of the sugar mill that we're using. On the left-hand side, you will have the ProFOS system continually scanning the bagasse as it goes through. We scan every 15 minutes. We've collected 6,000 spectra of bagasse over the last few months. And here you see some of the spectra we've collected. You can see they're a lot nicer than you get in the previous slide from the older system. And then the final part of the project is we're going to examine whether this works. At the moment we're collecting a huge number of samples, analysing them in the laboratory and building NIR models for those. And we will determine a report for a business plan to utilise this system not only in a sugar mill itself but also in other biorefineries processing different feedstocks. And I think that's where the most value will come from NIR. If you have seasonalities in your feedstock, you can discriminate between them and you can determine the composition of the materials going through using NIR spectroscopy. So to summarise what we've done, we've analysed a large number of samples via wet chemical methods. Anyone who's interested can go to the Dibanet website and download the analytical database that's there. We've improved the state of the art in the near-infrared analysis of biomass, lignocellulosic biomass, by allowing wet biomass to be scanned and its composition to be predicted with accuracy. We've launched a spin-out company which we will offer biomass analytical services, both using wet chemistry and NIR spectra. And we are applying near-infrared spectroscopy and demonstrating it as an online analysis tool that can be used in the future industries of second-generation biorefineries. We've also developed strong links between Europe and Latin America, and we will maintain these in the future. Thank you for your attention.
I'd like to open the questions, please.
Thank you very much for a very interesting presentation, especially the part on the in-line, at-line analysis using NIR spectroscopy. And yeah, it is true, it's a breakthrough if you can have this online analysis. Now, some technical questions probably we didn't have the time to elaborate. In the chemometrics, do you use principal component analysis in analyzing or getting these calibration curves? Or you use any other multivariate statistical analysis?
Partial least squares.
Partial least squares.
Yeah.
And just to continue, regarding the calibration and the transfer of calibration curve from laboratory to plant, or from one plant to another, or from another continent, or from one country to another, what is your experience with respect to transferability of all these calibration models?
What I didn't mention is that FOSS Analytical is also a partner in the Dibanet project, and they're working with us on Work Package 2. And we use their devices both in the lab and the online system. And at this very moment, next week, we are going to transfer the models that I developed in the laboratory onto the ProFOS online system. It's certainly possible. It requires some modifications, but it can be done. The one other point is that the Prophos uses a limited wavelength region between 1100 and 1650 nanometers, whereas the system that I used in the laboratory was between 400 and 2500 nanometers. Now, you need to still, if you're reducing the wavelength region, you have to have the important properties to be predicted in that limited region. And I've developed models using that limited wavelength region in the laboratory, and they can still predict glucan, xylan, place-on lignin, and these other components.
So in the real-time application, because you have some noise, do you get some kind of average signal in order to extract information?
We collect, for each spectrum that we get in the online system, it takes about 50 seconds, and that's an average of several hundred scans over that time.
Okay.
It's very quick to scan with the Prophos system because it's a diode array detector, there's no moving parts.
Okay, thank you.
Are there any other questions?
Just like for curiosity, what will be the natural competition for miscanthus in Ireland?
Combustion. It's used in a power station where it's combusted with heat. The point is that for combustion purposes, you want as dry as possible a biomass source. And that means you can only harvest it right at the end of that harvest window, so March and April. And as I showed there, you lose a lot of the biomass when you collect it at that point. If you have a Dibanet process, it's hydrolysis. So the water content, 50% that you have in the early window, is perfectly feasible for processing that in hydrolysis. So we can potentially lower the cost of the feedstock, or increase the potential revenues for the farmer in growing that feedstock, because he can sell more per hectare.
Social revenue? I mean, do you increase the amount of people that you need to process this thing?
What we would do is we would increase the interest in the crop, so we'd expand the number of hectares in production. And this is something that's particularly in Ireland is being actively encouraged by the government, moving away from pasture to some degree and moving towards energy crop production.
I mean, was there like some personal involvement in the discussions? I mean, like, did you have like a personal involvement on the discussions? Like, was it something personal? Or did you just...
I was looking for the best energy crop that's out there. And you have alternatives, you have reed canary grass, you have short rotation coppices, you have switchgrass. But the crop in Ireland, at least, that makes the most sense environmentally, commercially, is miscanthus. So that's why I selected it. We have analysed the other crops, by the way. You can download the data for these.
Is data instead of saying like you do something with this, you just have to go to the list or I could say like I mean, you've got a huge database, you've done like a huge amount of research. So I guess you could also like recommend to your clients.
We haven't analysed Greek samples. But in Greece, you will be more productive for miscanthus, providing you have enough water to crop. You have other materials that can grow in Greece, arundodonams and other things like that, which we can't grow in Ireland.
Are there any other questions? If not, I would like to thank Dan again for opening part of the
Just a final one. The ultimate objective with these online measurements which you have is to set up some optimization, real-time optimization policies with regard to the cross-operating conditions in the downstream process of your feed.
This is one point. You can also pay the supplier of the biomass according to the chemical value of the material. We find that with the gas, as an example, it doesn't vary that much, but the ash varies a lot. And if you're putting stuff that has a high ash content into your reactor, particularly if it's an acid hydrolysis reactor, you might need to modify the acid conditions to maintain your yields.
Correct.
So you can, what theoretically you could have is you could have a left and a right channel based on the scan, shift it one way, shift it the other, get 2 piles of biomass and use those accordingly, given the conditions.
Correct. Okay. Thanks again, Dan, for a very interesting talk.